Everyone please stand up as our VC arrives. May I kindly request our respected Vice Chancellor Ma'am, Professor Naima Khatun, to take her respective seat. And to our chairperson, Professor Mohammad Badruzama Siddiqui sir. Then our chief guest, Professor Andrzej Bajguz. Then our course coordinator, Professor Shamsul Hayat sir. Course co-coordinator, Qazi Fariduddin sir. And course local coordinator, Professor Vibha Sharma ma'am. रौनके बज़्म नहीं था कोई तुझसे पहले रौनके बज़्म तेरे बाद नहीं है कोई Assalamu alaikum and a very good morning ladies and gentlemen. With immense pleasure and great enthusiasm, I, Andli Banjum, your host for today's event, welcome you all with great joy and immense exultation. It is truly an honor to stand before such an esteemed audience invited by the pursuit of knowledge and scientific advancement. Today, we witness a significant milestone in the journey of academic excellence as the Department of Botany, Aligarh Muslim University, proudly presents one of its pioneering workshops under the Global Initiative of Academic Networks, a visionary program launched by the Government of India to elevate the quality of higher education through global collaborations. This workshop is not just a confluence of scholars, researchers, and experts. It is a gateway to innovation, a platform where ideas bloom, and a step forward in redefining research and learning. As one of the first ever Gian workshops conducted since the inception of this initiative, it marks a moment of pride and progress for our university, setting the stage for groundbreaking discussions and transformative insights. With this spirit of inquiry and excellence, let us embark on this enlightening journey together. तो अपनी देरीना रिवायात को बरकरार रखते हुए हम अल्लाह के कलाम के साथ आज के इस प्रोग्राम का आगाज करेंगे. And for this, I would like to call Inam to recite a few verses from Holy Quran.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم وهو الذي أنزل من السماء ماء فأخرجنا به نبات كل شيء فأخرجنا منه خضرا نخرج منه حبا متراكبا ومن النخل من طلعها كنوان ودانية وجنات من أعناب والزيتون والرمان مشتبها وغير متشابه انظروا إلى ثمره إذا أثمر وينعه إن في ذلكم لآيات لقوم يؤمنون صدق الله العلي العظيم. And he is the one who sent down rain from sky, causing all kind of plant to grow, producing green stalk from which we bring forth cluster of grain. And from palm trees, cluster of dates hanging within reach. There are also garden of grapevines olives, and pomegranates. Similar in shape but dissimilar in taste. Look at their fruit as it yields and ripens. Indeed, in these are signs for people who believe. और वही है जिसने आसमान से पानी बरसाया, फिर उसके ज़रिए से हर किस्म के नबातात उगाई, उससे हरे हरे खेत और दरख्त पैदा किए, फिर उनसे तह बतह चढ़े हुए दाने निकाले, खजूर के शगुफ़ों से फल के गुच्छे पैदा किए जो बोझ के मारे झुके पड़ते हैं। जैतून और अनार के बाग लगाए जिनके फल एक दूसरे से मिलते जुलते भी हैं और फिर हर एक की खुसूसियात जुदा जुदा भी हैं। ये दरख्त जब फलते हैं तो उनमें फल आने और फिर पकने की कैफियत ज़रा गौर की नज़र से देखो। इन चीज़ों में निशानियां हैं उन लोगों के लिए जो ईमान लाते हैं।
Thank you, Inam. May these divine words guide us towards wisdom and enlightenment throughout the workshop. गुलों में रंग भरे बादे नौ बहार चले, गुलों में रंग भरे बादे नौ बहार चले, चले भी आओ कि गुलशन का कारोबार चले. So as a token of our deep appreciation and gratitude, we now present welcome flower, or you can say bouquets, to our distinguished guest. And to do the honor, I would like to call Samin Zaidi to present the honor to our chief guest, Professor Andrzej Bajguz. Everyone, please give a big round of applause. And then I would like to call Bisma Hilal to present the bouquet to our respected Vice Chancellor of the University, Professor Naima Khatun, ma'am. Then I would like to call Sonia Wakar to present the bouquet to our Chairperson, Professor Muhammad Badruzama Siddiqui, sir. Then I would like to call Saida Zaima Zeib to present the bouquet to our course coordinator, Professor Shamsul Hayat, sir. Then I would like to call Sabaha Tehseen to present the honor to our Professor Vibha Sharma, ma'am, the local course coordinator. And then I would like to call Ms. Sana to present the honor to our course co-coordinator, Professor Qazi Fariduddin, sir. A small gesture, yet a reflection of our immense respect and administration. It is my pleasure to introduce our chairperson, Professor Muhammad Badruzama Siddiqui, sir, who joined Aligarh Muslim University in the year 1979 as an undergrad student and completed his entire higher education till PhD, making a lasting impact on our academic community. Initially specializing in plant taxonomy, sir later expanded their expertise to encompass allelopathy and environmental botany. Throughout this impressive career, Sir guided 12 PhD and 2 MPhil scholars. These scholarly contributions include over 150 publications in both national and international journals of high repute. हौसला मेरा ना नापो वक्त की रफ्तार से साल भर में एक सदी का काम मैं कर जाऊंगा. So with big round of applause, let's welcome our chairperson, Professor Muhammad Badruzama Siddiqui, sir.
Assalamu alaikum warahmatullahi wabarakatuh. Honorable Professor Naima Khatun, Vice Chancellor, Aligarh Muslim University, Aligarh. Chief guest, Professor Andrzej Bajguz, University of Bialystok, Poland. Guest of honor, Mohammad Azhar Shams. He's not here, I think. Then Professor Vibha Sharma, Local Gian Coordinator. Professor Samsul Hayat, Coordinator. Professor Qazi Fariduddin, Co-Coordinator. Teaching and non-teaching staff, members, research scholars, and students. Distinguished guests, ladies, gentlemen, a very good morning to all of you. Indeed, it is a matter of profound honor for me to express my heartfelt gratitude to you for being with us on this occasion. I am especially intended to Professor Naima Khatun, Vice Chancellor of our alma mater, an esteemed guest for sparing your precious time for the program which is being organized as an event of the centenary celebration of the department. On behalf of all the members of the department, I welcome you all at the inaugural session of this workshop. I feel immense pleasure in presenting before this galaxy of distinguished academicians and scholars a brief introduction of the Department of Botany. The Department of Botany was established in 1923 at the Aligarh Muslim University, stands one of the institution's most distinguished academic and research centers. With a legacy spanning over a century, the department continues to be hub for the excellence in the plant physiology, plant pathology, environmental botany, cytogenetics, plant breeding, plant biotechnology, systematics botany, and allelopathy. As a testament to its commitment to cutting edge, the research and global collaboration, the department is hosting the global initiative of the academic network, that is the Gian 2025. The course titled Advanced Course on the Determination of the Plant Hormones, Agriculture and Biotechnology from February 10 to 14, 2025. This prestigious event undergoes the department's vital role in the advancing scientific knowledge and fostering international partnership. The academic excellence, research contributions, and department offers undergraduate, postgraduate, and postdoctoral program supported by the distinguished faculty members, 10 professors, and 8 assistant professors with 36 staff members. Over the decades, it has nurtured 4 DSCs, 437 PhD scholars, and significantly enriching the field with over 4,500 research papers and 60 books. With a strong emphasis on the innovation and knowledge dissemination, the department regularly organizes seminar, workshop, and outreach programs, fostering academic discourse and interdisciplinary collaboration. It has successfully secured substantial funding from the premier agencies, including University Grant Commission, a special assistance program. The Department of Science and Technology Fund for the Improvement of the Science and Technology Infrastructure, that is the DST-FIST. The Department of Biotechnology boosts to University Interdisciplinary Life Science Department for the Education and Research, that is the DBT Builder. Recognition and global impact. The department takes immense pride in the achievement of the faculty, many of whom have received prestigious awards such as INSA Young Scientist Medal, Professor Hilala Chakravarti Award, Vigyan Ratan Samman, Panchanan Maheshwari Gold Medal. Furthermore, the several faculty members rank among the top 2% of the Indian scientists in plant sciences, as per the Stanford University study. The department's illustrious alumni have secured esteemed positions in the worldwide, reinforcing the global reputation for the academic and research excellence. As the department proudly hosts the Gian 2025, the course, we extend the warm welcome to all participants. We are confident that the insightful lectures, discussions, and collaborative engagements will offer the transformative learning experience, fostering new perspectives and advancement in the plant hormone research, agriculture, and biotechnology. Thank you so much.
Thank you so much, sir, for your valuable insights and encouragement. So, ladies and gentlemen, now it's time to introduce our course co-coordinator, Professor Shamsul Hayat, sir, a distinguished professor in the Department of Botany at Aligarh Muslim University, a recipient of prestigious awards like the Professor Hiralal Chakravarti Award and Dr. P.S. Khankoje Gold Medal. He has also been recognized as an associate of the National Academy of Agricultural Sciences with over 190 research papers and an H index of 70. His contributions to plant science are truly remarkable. Please give a big round of applause for Professor Shamsul Hayat, sir.
Respected Vice Chancellor, Ma'am, Chief Guest, Chairperson, Local Gyan Coordinator, faculty members, esteemed guests, distinguished speakers, students, and all attendees of the Gyan course, good morning and Assalamu alaikum. With great pleasure and sense of shared response, I welcome you to Gyan 2005 course on Advanced Course on Determination of Plant Hormone, Agriculture and Biotechnology, hosted by the Department of Botany here at Aligarh Muslim University and sponsored by IIT Hyderabad as well as the Government of India. Today, we gather within an institution dedicated to academic excellence and pioneering research to explore a topic that blends advanced knowledge on plant hormone. This course, offered under the Gyan 2025, has been designed to provide an in-depth understanding of plant hormones, their extraction, profiling, and applications in agriculture and biotechnology. The course combines theoretical lectures with hands-on tutorial to equip participants with both knowledge and practical skills in plant hormone research and with application with the set objective to explore the advanced techniques for the extraction, isolation, and profiling of plant hormone, to understand the role of phytohormone in plant growth, development, and stress response, to apply phytohormone to improve crop productivity under natural and stress, to familiarize participants with modern analytical tools and techniques used in plant hormone research, and the last objective that to discuss emerging trends and challenges in plant hormone biotechnology. The course is divided into 15 lectures and 5 tutorials covering both theoretical and practical aspects of plant hormone research. This course offers a unique opportunity to learn from experts in the field and gain practical skills essential for cutting-edge research in plant hormones and their application in agriculture and biotechnology. Therefore, this will give an immense skill to our students. Aligarh Muslim University has been a center of advanced research, and it is our honor to continue its tradition today. Our department is committed to advancing knowledge that furthers scientific understanding, serves the community, and contributes to sustainable development. By the end of course, participants will gain a comprehensive understanding of plant hormone biology and its application, acquire hands-on experience with advanced analytical tools and techniques. I appreciate your commitment and welcome to the Department of Botany, Aligarh University. Let us make this Gian program a meaningful and transformative experience. Thank you and welcome to the Gian course.
Thank you so much, sir. So, ladies and gentlemen, it's my immense honor to introduce a distinguished academician, researcher, and administrator with over 3 decades of experience. She has served as professor and chairperson of the Department of Psychology, principal of Women's College, and now the vice chancellor of the Aligarh Muslim University, Professor Naima Khatun. She has authored 6 books, supervised 15 PhD scholars, and delivered lectures at top global institutions. A dynamic leader, she has held key administrative roles, including provost, deputy proctor, and deputy director of RCA. A true inspiration, Professor Naima Khatun ma'am is a proud of Aligarh Muslim University, twice elected to the Women's College Student Union and recipient of the Papa Mia Padma Shri Award of Best Girl.
Assalamu alaikum and a very good morning to all of you. The chief guest, Professor Andrzej Bajguz from Poland, chairman of the department, Professor Badruzzama Siddiqui, the course coordinator, Professor Shamsul Hayat Saab, and the course co-coordinator, Professor Qazi Fariduddin, and of course the local coordinator of Gian, Professor Bibha Sharma, and all the faculty members sitting here and the students and researchers. A very good morning to all of you. It is indeed a great pleasure for me to be here this morning. I am sorry I could not, I will not be able to stay here for long because I have a scheduled meeting already. But I am happy that this Gian program is being very well taken, and participants have come from many universities. As it is, the program introduced by the Government of India Ministry of Education is indeed a very useful program, because the trends in the various disciplines, what is going on in the prestigious universities all over the world, that is why it has from international experts in the field will come, and that they will give their inputs to our researchers. So it is a very good academic interaction among the researchers and among the faculty members. And I wish and I hope and I urge all the research scholars, please do attend all the lectures that the various resource persons are going to deliver. Because sometimes you have some other preoccupation, but if you attend this lecture, that is going to be very, very beneficial for all of you. This is particularly for the research scholars and even for the master's students. And as it is, the Department of Botany has a very glorious past. We all know that in the past several years, the Faculty of Life Science in the discipline, we are #1 in the ranking, or 1 or 2 always. And the life science, the contribution of botany, zoology, and biochemistry, we all know, and the research publications, and the impact factor, and all those those points that are required for international ranking. So the contribution of the Department of Botany is very, very remarkable. So I congratulate the chairperson and all the faculty members and all researchers who have contributed it to the discipline. And as you all know, it's a very old department, and very renowned scientists from this very department have their names in. I think that Janki Vallabh award was from one of the, Professor Vashni, I remember, was a student of this department. Which award it was, Qudsiya? Janki Ammal. I said it is, well, it is Janki Ammal Award. All academies also, yes. Vashni, Professor Vashni, yes, I remember, very young researcher. So this department has many credit to its crown, and it is indeed, it is very satisfying and very fulfilling when the young researchers and the faculty members contribute. And so Aligarh Muslim University is also a very old department. We started in 1875, and as a university by an act of parliament as a federal university, we started in 1920. So it is more than a century old. And we have our uniqueness in the university, though it is not a very big city, it's a small, you can say a sleeping educational town. Not with the hustle bustle of big cities. And I hope you will have time to visit the campus and see our university. And I'm sure with your input and with your knowledge, our students and faculty members will immensely be benefited about it. And I once again congratulate the department for organizing this seminar, which is truly academic. Thank you so much.
Thank you, ma'am, for gracing this event with your presence. मैम वैसे तो वक्त नहीं है पर फिर भी दो लाइनें कहूँगी. हम नहीं बादे सबा तेरे करम के मोहताज, हम तो खुशबू हैं बिखरने का हुनर आता है. And with this, ladies and gentlemen, as a mark of appreciation, we now present mementos to our distinguished guest. And for this, I request our chairperson, Professor Mohammad Badru Zama Siddiqui, sir, to present the memento of honor to our respected Vice Chancellor, ma'am, Professor Naima Khatoon. Everyone, please give a big round of applause. Next, I request our Vice Chancellor, Professor Naima Khatoon, ma'am, to present the memento to our chief guest, Professor Andrzej Bajguz. Then I request Professor Vibha Sharma, ma'am, to present the honor to our chairman, Professor Mohammad Badru Zama Siddiqui, sir. Everyone, please give a big round of applause. Then I request Professor Qazi Fariduddin, sir, to present the honor to Professor Vibha Sharma, ma'am. Then I request Professor Aqil Ahmad, sir, to present the honor to Professor Qazi Fariduddin, sir. Then I request again Professor Aqil Ahmad, sir, to present the honor to Professor Shamsul Hayat, sir. Thank you, everyone. Now it's time that we reach a moment of great honor, the Felicitation of Lifetime Achievement Award.
So we have a few awards. This is one of the Lifetime Achievement Award. A disciplined and dynamic man who has taught plant physiology during his entire career, is one of the best teachers. He joined this university as a graduate student and completed his PhD in the year 1976. He was appointed as a lecturer in 1977 and later promoted to reader and professor in the department. He joined as a postdoctoral fellow on Danida Fellowship in Denmark. After coming from the Denmark, he started working on plant hormone. So the work on plant hormone is started by the person. He is one of the pioneer research of the plant hormone in the AMU. Since then, so many students have been awarded doctorate degree in plant hormone. He has guided a number of PhD students and published more than 200 research papers in the journal of repute. It is our pleasure to honor him with a Lifetime Achievement Award in an event based on plant hormone for which he is internationally acclaimed. Now I request the dignitaries on the dais to facilitate the award to Professor Aqil Ahmed. Sir, please come forward to receive the award. Yeah, one more. A person who has taught taxonomy, a tough subject of botany for the last 35 years, and has the administrative capability to shape the department in his chairmanship, guided to complete one of the successful national conference on medicinal plants after a decade, and also helpful in organizing this event. He joined the university as a graduate student and completed his PhD under the great taxonomist of India, Professor Wazahat Hussain. The field of his specialization and teaching interest is plant taxonomy. Professor Badruzzama Siddiqui produced 35 PhD in the field of plant taxonomy and ethnobotany and allelopathy. Today, we have honored him with a special award. So I request the person on the dais to facilitate the award to Professor Badruzzama Siddiqui.
Ladies and gentlemen, we are delighted to announce the release of the groundbreaking book, Strigolectrons, edited by Dr. Gausia Bashri ma'am, Professor Shamsul Hayat, and Dr. Andrzej Bajguz. This book introduces a newly discovered class of plant hormones, shedding light on their role in plant growth, development, and stress responses. With contributions from leading experts, it provides in-depth insights into their biochemical mechanisms and potential applications in agriculture and biotechnology. Everyone, please put your hands together for the book edited by all these three dignitaries, Strigolectrons. Please be seated, everyone. So now it's time to introduce our local Gian coordinator, Professor Vibha Sharma ma'am, a distinguished faculty member in the Department of English at Aligarh Muslim University, specializes in theatre and performance studies, translation studies and cultural studies. As a visiting faculty at Stockholm University since 2013, she has received international fellowships and presented research across Europe, Asia and South America. She serves as the Deputy Coordinator of the DRS UGC SAP Phase 2nd program on translation studies and actively mentors young scholars. So with big round of applause, let's welcome Professor Vibha Sharma.
Thank you for this generous introduction. A very good morning to all of you. Assalamu alaikum, adab. Madam Vice Chancellor, Professor Naima Khatun, in absentia, she was here with us a moment ago. Chief guest of today's program, Professor Andrzej, Chairperson, Professor Badruzama, Coordinator of this Gian course, Professor Shamsul Hayat, and Co-Coordinator, Professor Qazi Fariduddin. It is indeed an honor for me to be present in one of the most illustrious departments of my university, and I'm proud to assert this, underline this once again, that Department of Botany has been one of the front runners in ranking and has earned accolades for our university for consecutive years as #1 department in the entire country. So I think you all deserve a big round of applause. Congratulations for being part of this department as students, as teachers. This connection today that I have developed with this department is definitely a kind of achievement for me, because I am among the illustrious achievers in terms of research and academics. In my role as local coordinator of Gian, I must speak a few things about this very important initiative that Government of India has taken. And we cannot thank our seniors enough for this wonderful program. Global Initiative of Academic Networks means that we create a network with the global achievers in our distinguished fields, research fields, professors, scientists, academicians, artists. We need to connect with the entire globe as one big family of sharing and nurturing ideas for the next generations. And our country is not one of the developed countries, but the fact that we think of funding purely, 100% funding for such programs, and hundreds of such programs are running across the country, not just in my university, our university. So it is a big thing. We must make the best of it. To the students and research scholars, I must reiterate what the Vice Chancellor ma'am has said, that please don't take it for granted. You must take out time to be in the lectures, in the discussions as much as possible, because these are very valuable programs. These are being conducted with great effort, great initiatives, and a lot of hard work that each one of the stakeholders take. I'm happy. I must congratulate Professor Shamsul Hayat and the co-coordinator, Professor Qazi, because this is very important that somebody takes the initiative, they materialize it, and they convert it into a reality of what you see today. So this is one aspect of Gian. Um, you see the word Gyan strikes a chord because it means knowledge in Sanskrit. So, so the program's coinage is very, very meaningful in that terms also. The second thing I must take a moment is to express my ignorance amidst the galaxy of botanists present here. As I entered the department, I uttered a sentence that the subject has nothing to do with me. I wanted to actually say that I wish I could speak something in botany because I always speak in English. But I can say something in English, which is an underline for botany, that when earth has to laugh, it laughs through flowers. When the earth has to express its smile, it spreads through the fragrance of flowers, the smell of the trees, fruits, and of course the soil. Mahatma Gandhi had once said that if we are not able to dig soil and plant a seed and take its care, we are not humans. And I think botany underlines this fact. Today we are living in 21st century, lives of apartments and high-rise buildings, and we love having greenery in our apartments. We bring flowers, we bring flower pots, we gift that. But I think it's more important to take care of the trees, the bigger plants, because that's where the real powerhouse of green energy lies. And I must share with the students of botany here that there is a beautiful poem in our English literature which we teach to students. There was a great poet called Matthew Arnold. When his friend died, he lost his young friend. He was a young man like you. He lost his young friend who was a university mate in Oxford. Oxford University and Cambridge University are our role models in terms of structuration, because our founder, Sir Syed Ahmad Khan, had created the structure of this university not just ideally, but also in terms of its physical beauty. And so the plants that we have on this campus are its decoration that was envisioned by Sir Syed Ahmad Khan. In that poem, the friend who loses his friend writes a poem, which is a long poem, and in every stanza, there is a mention of flora. He mentions that how me and my friend used to take visit in the campus, we used to walk, and how we used to come across flowers and trees. So this poem has actually become a documentation of the beautiful university campus, and half of those flowers do not exist today because this poem was written in the 18th century. So I'm asking you to please open your gates to literature students. One of the literature students adopted botany as an open elective, and I encouraged him because many of the teachers were saying that it has nothing to do with literature. So with FIUP, with NEP, we have options to collaborate more and more. Who knows, one of your friends can also write such a poem, but obviously with your help. So we need to collaborate more and more. Botany is not away from literature, because when this program began, the first thing was a poetry that was recited. One was in the praise of the Almighty, and one was in the praise of the guest, and that is literature. So literature cannot escape from a botany classroom, and botany is, of course, all around us. We breathe botany, we breathe the fragrance of flowers and the planet Earth. So let's keep our planet Earth more smiley, more happy, and let us collaborate more. That is very important. So this is my message, and this is the message of Gian also. This is the message of NEP also. Thank you very much. I wish you all the best for the next few days. Make the best of it, and please attend all the lectures. We'll meet again in the valedictory to know what has gone through. Thank you very much.
Thank you so much, ma'am, for such thought-provoking words. तो प्रोग्राम को आगे बढ़ाने से पहले मैं अपने कोऑर्डिनेटर्स प्रोफेसर शमसुल हयात सर, प्रोफेसर काजी फरीदुद्दीन सर और हमारी लोकल कोऑर्डिनेटर प्रोफेसर विभा शर्मा के लिए 4 लाइनें कहना चाहूँगी। शबनम की नमी पाओगे कांटों की चुभन में, अंगारों का अंदाज गुलाबों में मिलेगा। मुझको ज़रा पहचान लो ऐ आज के लोगों, हर नक्श मेरा कल के निसाबों में मिलेगा। So ladies and gentlemen, with this, now it's time to introduce our chief guest, Dr. Andrzej Bajguz, an eminent scientist and Dean of the Faculty of Biology at the University at Bialystok, Poland. A visionary in plant biochemistry, his groundbreaking research on brassinosteroids and their role in plant stress tolerance has revolutionized our understanding of phytohormone interactions with over 120 publications, 4 books, and numerous accolades. He stands as a beacon of excellence in plant science, a mentor to many, an active contributor to prestigious scientific society, and a leader to academia. Dr. Bajguz's work continues to shape the future of plant biology. Everyone, please give him a big round of applause.
Thank you very much. Respected professor, doctors, and students, workers of Aligarh Muslim University. Good morning, everyone. Salam alaikum. I would like to thank authorities of this university, especially Professor Shamsul Hayat, my best friend, Indian friend, for inviting me to present some lectures on extraction, on determining plant hormones, and some news about brassinosteroids, phytohormones, melatonin, and strigolactons. So I'm honored that it's the first time I am in India. This is the fine trip to this beautiful country. So I'm very glad that I'm here. I'm on visit Aligarh and other places. Thank you very much once again. Thank you, Shams.
Thank you so much, sir. Well, he'll not be understand the Urdu shairi, but still I'll recite 4 lines from English. With wisdom vast and vision bright, you grace our day as a guiding light. A guest, a guide, a shining flame, we honor you in heartfelt name. Thank you, Professor Bajguz, for gracing this event with your presence. Since every successful event is the result of collective efforts, so as we are near the conclusion of this formal session, I would like to invite our course co-coordinator, Professor Qazi Fariduddin sir, a distinguished professor of botany at Aligarh Muslim University with extensive research on plant growth regulators and their role in stress tolerance. He has contributed over 90 research papers to reputed international journals. A Boy Scout and Raman Fellow, he has collaborated with leading global institutions and successfully led multiple funded research projects shaping the future of plant physiology and biochemistry. So everyone, please give him a big round of applause.
Respected Vice Chancellor, Professor Naima Khatoon, Professor Andrzej Bajguz, Professor Aqil Ahmed, Local Coordinator of Gian, Professor Vibha Sharma, Chairperson Department of Botany, Professor Badruzzama Siddiqui, distinguished guests, dear participants and friends. It's my privilege and honor to propose a heartfelt vote of thanks on this auspicious occasion of the inaugural of Gian course titled Advanced Course on Determination of Plant Hormones, Agriculture and Biotechnology. First and foremost, I would like to extend my deepest gratitude to our chief guest, Professor Andrzej Bajguz, a distinguished foreign faculty member, for gracing this event and sharing his invaluable insights and expertise. Your words have not only inspired us, but have also set a tone for the days of learning and collaboration ahead. I would like to express my sincere thanks to Professor Naima Khatoon, our esteemed Vice Chancellor and the patron of this course, for her unwavering support and motivation, which have been instrumental in the successful organization of this event. I would like to acknowledge Professor Aqil Ahmed, whose guidance and mentorship have shaped and carved a niche for me and Professor Shamsul Hayat in academic world. Sir, your legacy continues to inspire and energize us in our endeavors. We are immensely grateful to Global Initiative of Academic Networks under the Ministry of Education, Government of India, for funding this incredible platform. This initiative has brought together experts and participants from across the globe, fostering knowledge, innovation, and collaboration in the field of plant sciences. I special thanks to Professor Anwar Shahzad, MIC Land and Garden, for making this event very wonderful. And I must also thank local coordinator, Professor Vibha Sharma, who has recently assumed this challenging responsibility and has worked diligently to ensure the success of this event. I would like to acknowledge unwavering support of Chairperson, Professor Badruz Zama Siddiqui, Department of Botany, our university administration, and all the faculty members who have contributed to the smooth functioning of this event. Last but certainly not least, I extend my heartfelt gratitude to the technical and support staff, volunteers, everyone behind the scenes who have worked tirelessly to ensure the success of this inaugural function. Resource person and dear participants of the course, as we embark on this journey of learning and discovery, let us remember that the knowledge we gain here has the potential to transform agriculture and biotechnology, paving the way for a sustainable and prosperous future. Once again, thank you all for being part of this momentous occasion. Let us make this course a memorable and impactful experience for everyone involved. Thank you, and maybe the days ahead be filled with fruitful discussion, innovative ideas, and a collaborative spirit. Thank you.
Thank you, sir, for acknowledging the efforts of everyone who contributed to making this event a success. And for everyone behind this event, I would just like to say, कुंदन हुए हैं आग भी अपनी लगा के हम पर किसी सुनार की मर्जी नहीं चली. तो ख्वातीन और हजरात, अलीगढ़ मुस्लिम यूनिवर्सिटी की ये रवायत रही है कि किसी भी प्रोग्राम का इख्तिताम अपनी मादरे दर्सगाह के तराना और नेशनल एंथम के साथ करते हैं. सो आई रिक्वेस्ट एवरीवन टू स्टैंड व्हेन नेशनल एंथम बिगिंस. And I request the volunteers to join to the podium for Tarana. Tarana है ना?
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Thank you everyone. With this, we conclude the inaugural session of the Gian workshop. So I invite you all to join us for high tea in the department lawn.
Hello, 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 hello.
Hello and welcome to all. Good afternoon. So, we are assembled here for our the first lecture in relation to the advanced course on determination of plant hormones, agriculture and biotechnology. This course is offered under the Gian platform, that is basically global initiative of academic network. This is the program which is basically initiated by the government of India. Earlier it was run by the IIT Kharagpur. Now it is under the IIT Hyderabad. Basically, this course is designed to provide a comprehensive understanding of various plant hormones, their critical role in plant growth and development, and their applications in modern agriculture as well as technology. As most of the people who are sitting here or who are on the online, they may know about the plant hormone. It is also termed as a phytohormone. They are the key regulators of physiological processes in plant that has basically influenced everything from the seed germination to the growth as well as flowering to the crop productivity, both under normal as well as stress conditions. We have some objective behind this course. That first is that we have to explore the techniques for the extraction, isolation and profiling of plant hormone. So, this is the first objective of our course. Then we have the second objective in mind, that is to understand the role of phytohormones in plant growth, development under normal as well as stress conditions. The third objective is, which is basically more fruitful in relation to the farmers, that to apply the plant hormone by various methods for improving the crop productivity under normal as well as stressful conditions. To familiarize participants with modern analytical tools and techniques used in plant research, that was another objective. And the last objective is that to discuss emerging trends and challenges in plant hormone biotechnology. So, with these objectives, a total number of 15 lectures and 5 tutorials will be covered. In that, the lectures will be based on the theoretical knowledge and the tutorials will be related to the practical aspects of the plant hormone. Okay. So, here we are having the lecture details, that what are the lectures will be covered in all the 5 days. The program of the event has already been shared on the WhatsApp group, that on which days, which lecture at what time will be there. So, the first lecture is basically related to that of the extraction and isolation of phytohormones from plant tissues, that is from past to present. क्या हुआ last and now. In this lecture, some of the major points which will be covered is the overview of historical and modern methods for extracting and isolating plant hormones. Besides that, what are the challenges and advancement in extraction technique for the plant hormone will also be discussed in this lecture. Besides that, there is the various chemical structure of phytohormone which has been isolated by the various techniques is also the part of the lecture. Then we have the second lecture, that is hormone profiling in plant tissue. In this lecture, some of the major themes which has been covered are, that is, techniques for identifying and quantifying plant hormone. And then we have importance of hormone profiling in understanding plant physiology. These lectures will be mostly covered by the Andrzej, and in total he will deliver the 9 lectures in the 5 different days. Okay, so this is one of the lecture which will be delivered by him. Then we have the advantages of isotope labeled standard, as well as the limitation and quality control that will also be covered under this lecture. Then we have the third lecture, that is application of the various phytohormones in increasing the crop productivity in natural and stressful conditions. We have various type of stresses in the environment, that is salinity, heavy metal, nowadays nanoplastic, microplastic, and so on. So many things are there that will be covered. So the points which will be covered are that role of phytohormone in enhancing crop yield under stress, then the case studies on the use of hormone in agriculture, then the advances in phytohormone research and technology for the sustainable agriculture. And another major point, that the comprehensive understanding of the phytohormone mechanism for addressing global food security. It means that the phytohormone have a major role in relation to the global food security. Then the fourth lecture is basically, I will give the lecture on this particular topic, that is the regulation of photosynthesis by brassinosteroids. As you all know that brassinosteroids is basically a new class, or you can say the 6th class of the plant hormone, and at present in Aligarh itself, we both, Professor Qazi Faruddin and myself, have worked a lot on this particular hormone. So most of the study in relation to the regulation of photosynthesis by brassinosteroids will be the case study which has been completed in our lab. So in that, we will discuss the impact of brassinosteroids on photosynthetic efficiency. Most of these parameters is generally monitored by the LICAR photosynthetic system which we have, in relation to that, we also have a tutorial, and that on that tutorial, Dr. M.C. Arif will give you some handouts. Then in the second point, we will discuss the mechanism of brassinosteroid mediated regulation. And in the last, we have also discussed the interactive effect of brassinosteroids with other hormones. In the fifth lecture, that is basically related to that of the methods for modeling phytohormone mediated signaling in plant development. This lecture will be covered by Andrzej. In that, he will discuss the overview of phytohormone signaling, computational and experimental approaches to model hormone pathways. Then what are the applications in understanding the plant growth and development, as well as there is some challenges in relation to that, as well as that of the some benefits in phytohormone mediated signaling. The last, which will be the part of that, the integrative framework of most of the hormones, as all of you are knowing, basically up to 5 or 6 hormones, but apart from that, the other hormone will also be discussed. Then we have the sixth lecture, that is techniques used for the characterization of the stress markers in plants exposed to heavy metals. There is a various analytical methods for detecting stress markers in plants that will be discussed. Besides that, what are the roles of phytohormone in relation to heavy metal tolerance will also be part of this lecture. Then we have the another point which will be under consideration in this lecture is that developing heavy metal resistant crop via genetic engineering. This point is required as most of the farmers are facing problem in relation to the heavy metal. So we have to develop something like that, that the crop should not be damaged. And a combination of biochemical, spectroscopic, molecular and imaging techniques is also essential for studying various plant stress markers that will be shared in this lecture. Then we have the seventh lecture, that is polyamines, that is also under consideration as a new plant hormone. That the topic is, that is polyamines that allies in plant resilience against abiotic stress. This lecture will be given by Professor Qazi Fariduddin. His most of the research is also on this particular area. In that lecture, he will describe what is the history of the polyamines, then some of the functions of polyamines under natural as well as stress conditions, and he will also discuss that is there any stress tolerance mechanism in relation to polyamines or not. Besides that, a mechanism of polyamine mediated stress response will also be shared, and the last one that will be the part of that lecture is that what are the role of polyamines in the growth and development of the plant. So, in that we have a number of case studies which has been conducted in our lab under the supervision of Professor Qazi Fariduddin, and that will be summarized in that particular lecture. Then we have the another lecture that is related to that of the challenges and practical problems of plant hormones in biotechnology. So, in this lecture, a limitation, always there might be a limitation if there is some practical problem. So, we have a number of limitations because it is not possible to do whatever we want, because there is sometimes there is an environmental problem, then there is sometimes various suppose in our case in Indian culture, that if we are doing some experiment on the GCMS or the LCMS and light is switched off, then we have a no complete current. So, these are the some limitations are there. So, limitation as well as that of the ethical consideration in plant hormone applications will also be discussed in this, as well as that one of the more important points which is directly related to the genetic engineering or biotechnology, that what will be the future direction of the plant hormone and what its role in relation to the biotechnology, that will be discussed by the Andrzej. Then we have the identification of the stress marker in relation to hormone and biotechnology. Besides that, nowadays everyone is talking about the nanoparticle. In every lab, the people are working on the nanoparticle. So, there might be some correlation between the nanoparticle and phytohormone or not. It will also be discussed in that particular lecture, that what are the role nanoparticles have in relation to the phytohormone in biotechnology. And the latest one which is under consideration in most of the laboratories of the China, that is microplastic and nanoplastic. They are working in there as well as that of the UK, Poland, Russia and so many countries nowadays. Microplastic and nanoplastic interactions are discussed. We have also published one review article with Andrzej on the microplastic. So, what are the interaction with the phytohormone with these micro and nanoplastic, that there is some advantages or disadvantages, what are there, that he will discuss in that particular lecture. Then we have the one of the lecture, that is the around the brassinosteroids in algae. This is one of the, I think, the favorite topic of you. It is one of the favorite topic of Professor Andrzej, and he has a command on this particular topic, and most of his research is also related to that of the algae and brassinosteroids. So, you can say that he is one of the best to deal with that of the brassinosteroids in algae. So, in that he will discuss about the role of brassinosteroids in physiology of algae. Then we have a comparative analysis with higher plants, that what the role the algae have and what the role the higher plant have. He will give a comparative analysis between these two products. Then we have the also in this lecture, that what are the ecological significance of algae in relation to brassinosteroids, whether is there any detection of challenges of brassinosteroids in algae will also be covered in this lecture. And in the last, he will give an overview of biosynthesis, BR biosynthesis in algae. That is the overall theme of that particular lecture. This is one of the totally new hormone, that is keratin. And in this lecture, one of our retired colleague, Professor M. Masroor Khan, who has worked a lot on this particular hormone and also developed one of the instruments that how this keratin is going to be, you can say the isolated, or you can say the produced. So he has made that particular instrument that is also available in our lab. So he will discuss its origin and overview of the keratin. So he is the pioneer in this department about the keratin. So he will the most ideal person to discuss about the keratin overview as well as that of the origin. Then what are the signaling pathways are involved in the keratin? He will also discuss it. And besides that keratin, there is some other growth promoter which have a role in the growth and development of the plant will also be the part of his lecture. In that one, he has developed that how the production of methanol is boosted by some novel technique that he has produced that particular technique. And in the same lecture, he will also discuss the role of chitosan and its derivatives on the plants as well as that of the plant parts. Then we have another lecture, that is the role of hydrogen peroxide in plants under abiotic stress. This lecture will be given by Professor Qazi Fariduddin. On this hydrogen peroxide, he has produced a number of PhD studies, and so he is the ideal person here to discuss the role of hydrogen peroxide. So in his lecture, he will signify the dual role of hydrogen peroxide as a signaling molecule as well as that of the stress marker. In detail, he will discuss about this. Then what are the mechanisms that has been involved in the hydrogen peroxide mediated stress response will also be covered in his lecture. And overall, in the last, he will summarize that what are the role of hydrogen peroxide in growth and development of the plant. In his lab, a number of plants has been studied under the hydrogen peroxide. So the case studies with relation to that of the hydrogen peroxide will also be shared on the screen in his lecture. Then we have another lecture that will be given by the Andrzej, that the melatonin and strigolactone, that is a new types of phytohormone. On melatonin, I and Professor Fariduddin is also working, and in the inaugural you have already seen that all the three editors who have edited the strigolactone book are here, Andrzej, myself and Gausia. So we have also started on this particular hormone. It is considered as a because this melatonin and strogolectin both are considered now as a new type of phytohormone. A number of studies has been done which considered is under the category of the plant hormone. In that, the Andrzej will discuss about the functions of melatonin and strogolectin in plant growth under normal as well as stress. Because in the normal there is some other response, and in the stress the response will be different. Then he will also cover the recent discoveries and its application. And the another major point is that signaling pathway of both strogolectin and melatonin. So these are the some of the major points which will be covered in the lecture 12. Then we have another lecture, that is phytohormones. That lecture in this, the Professor Andrzej will discuss about the phytohormones. That is a totally new, and I think we have a not much more role, but my group and his group has already published one review article on the phytohormones. So some of the studies is common on this particular area, that the overview of phytohormones and their role in plant defense and growth. These will be discussed. Then this particular hormone have any potential implication in relation to agriculture or not, he will also discuss in detail. Then we have a structural similarity to insect molting hormone. That what are the insect similarity between phytohormones and the insect molting hormone will also be discussed in detail. Then we have the challenges and opportunities in relation to phytohormones. What are the importance of sustainable practices in relation to this particular hormone, which will also deal by this great man. So we have another theoretical lecture, that is regulation of nitric oxide and what are the role in agriculture. So nitric oxide is one of the signaling molecule on which a major studies are going on. This is also comes and considered as a new class of plant hormone. The lecture on this particular topic will be given by our young lecturer, that is Assistant Professor Dr. Gosri, who has done a lot of work during his PhD as well as after PhD on the nitric oxide. So he has one of the best man in the present legal condition to discuss about the nitric oxide. So in that particular lecture, she will discuss about what are the role of nitric oxide as a signaling molecule in the plant. In detail, she will discuss on this. Then what are the implications in improving crop productivity and stress tolerance. So then another point, what are the signaling pathways of nitric oxide generally in some of the plant, the signaling pathway may vary. As I have also guided 2 students for the PhD on the nitric oxide. So, I have also some knowledge about this, that on the basis of the genera as well as that of the species of the plant, the signaling of nitric oxide is generally differ. Okay. Then we have an interaction of nitric oxide with other plant hormone. This point will also be discussed in our lecture. So, these are the some of the lectures, theoretical lectures, which will be covered in these 5 days. Besides that, we have a 5 tutorial, that is one of the major part of this program. Generally, it is the theme of the Gyan, that besides the lecture, we should give a hands-on training which is available in your department in relation to the phytohormone. That will be also be the part of this program. So, in the tutorial 1, that will be taken place today after 3:30, that is hands-on training on water potential meter. This particular instrument we have, and we have our old student, now the assistant professor Dr. Yamshi Arif, who have a good knowledge that how to do it. She will describe its detail and give a hands-on training. It actually it is the we have to give a hands-on training in the field condition, but here we have a limitation of the Gyan, that each and every lecture has to be online, or you can say the live streaming should be there. So, this instrument is not possible to go there with that of the videography. Instrument can go there, but the whole videography system is not possible to shift to the field condition. So, that is why all the hands-on training we will provide inside this room mostly. So, in this tutorial, a practical session in relation to measuring water potential in plant tissue will be done, and then what are the application in understanding plant water relation will also be discussed by the Dr. Yamshi R. Then we have a tutorial, that is training on our gas chromatography and mass spectrometry. So, this will be delivered, the whole methodology I will deliver in this classroom, and after that by a batch of students, just we will go on to the our the instrumentation center, just where there in that we will just give you that how we can analyze the plant hormone under GCMS by a 10-man group. So, actually it is a very big and you can say the costly instrument, and required a lot of money for the purchase of the chemical to run that particular instrument. So, we have a limited fund in the Gyan, so it is not possible to do for each and every student and analyze by the GCMS. So, another, that what are the techniques for after just measuring that particular hormone, there is a technique that how the data which has been observed under this particular instrument can be interpreted, that will also be told to you in that particular tutorial section. you all will get it all lecture in your email as well as that of your in the WhatsApp group. I will share all the lecture, so you can have all the lectures there, and it is also available on the IIT Hyderabad site as well as also available in the YouTube. It is going on in the live as well. After the completion of the course, it will also be available. Okay, Hitesh? Yes. So we have another tutorial, that is the training on PAM, that is pulse amplitude modulation, PAM fluorometry. This we have just a new instrument we have bought by the finance of the Aligarh Muslim Ministry has given some of the money to purchase this particular instrument, and recently we have it, and this will basically provide good data and which relation to that of the photosynthetic efficiency. And so in this we have a practical session on measuring the photosynthetic efficiency using PAM. This particular instrument use the photosynthetic efficiency. Okay. Then we have a what application in studying hormone treated blood. So we can compare the results of the control and results of the hormone treated plant, that will be compared that, and we should find a difference between the control as well as that of the hormone treated plant, or as well as that of the under the stress conditions. Then that is the monitoring of the plant stress with the help of PAM. It is the easiest method. Without detaching the leaf, we can measure these parameters here. So there is no harm to the plant, and we can have this particular. Then we have another tutorial, that is, it is related to that of the tissue culture technology and plant hormone. Okay. So we will discuss the techniques of the plant tissue culture and hormone application. Then we have a case studies in relation to hormone mediated plant regeneration, that I will give you a brief introduction about this in the tutorial section here. Then we will go to the tissue culture laboratory. Myself or Professor Anwar Shahzad will give you that what are the techniques has been used in that particular area. Then the last tutorial we have, that is the most important. We have a wonderful instrument, that is the Leica photosynthetic system. It is at present, it has cost about 50 lakhs. At present, it cost about 50 lakhs, and it is not possible in the present condition to purchase such an instrument. We have the old one which we have been, but still, alhamdulillah, it is working. So the Dr. Yamshi Arif, who has a knowledge of just giving a training, so she will discuss the practical session using this Leica system to measure most of the photosynthesis. Besides that, what are the applications in studying hormone effects on plant physiology can also be studied by using these plant hormones. Okay, these are the different lectures and tutorial which will be covered under this Gian program. That will be most helpful parameters or most helpful topics are here in relation to the plant hormone, which will be very useful by all of you. So by the end of the course, we can say that the participants will gain a comprehensive understanding of plant hormone biology and its application. Then acquire hands-on experience with advanced analytical tools and techniques. So, this course basically offers a unique opportunity to learn from experts in the field and gain practical skills that are essential for your further research on the plant hormone and that their role in agriculture and botany. Thank you. Is there any question? These are the online. आज की रिपोर्ट भूल गया था मैं यहाँ की बैठे क्यों रिपोर्ट भी देख रहे हैं थोड़ी सी बना दो थोड़ी नो यू डोंट यू हैव योर लेक्चर एट 2:30 ओके देयर एनी क्वेश्चन तो करिए आप लोग अच्छे से अटेंड करें इट विल बी ए गुड कोर्स एंड यू विल गेट ए मच बेटर ओके नाउ वी हैव ए लंच एट 1:30 ओके 1:30 पे भी आप लोग जो लंच करेंगे ठीक यहाँ बंद कर दे लाइव तो हो गया आफ्टर लंच स्टार्ट करिएगा जी ठीक आइए घूम लीजिए फिर करके कराना है
welcome you all in the second session of the course on the plant hormones. So in the second session, the first lecture is given by our chief guest, that is Andrzej Bajguz, which is on the topic that is the extraction and isolation of phytohormones from plant tissues from past to present. So I welcome our chief guest Andrzej Bajguz to deliver his talk.
No, it will be fine when you stay in here. Good evening, everyone. I'm great pleasure to welcome you all to the lectures of extraction of and isolation of phytohormones from plant tissues from past to present. My name is Andrzej Bajguz, and I am honored to be a part of this global initiative of academic network course. When we dive deep into the advances methodology determining plant hormones, particularly in the context of agriculture and biotechnology. Let's begin by looking at the fundamental role of phytohormones in plant biochemistry and plant biology. Before we dive into the specific of extraction and isolation technique, it's important to understand why phytohormones are and why phytohormones are so crucial in plant physiology. Phytohormones are naturally occurring organic compounds that influence various physiological processes in plants. They play an essential role in nearly every aspect of plant life, including growth, development, and responses to environmental stresses. These hormones act as key signaling molecules, coordinating many biochemical processes, physiological and morphological processes too. The effect extend beyond individual cells. Phytohormones regulate nutrient allocation, determine the sourcing balance, and mediate plant responses to both abiotic and biotic stresses. Unlike in animals, where hormones are typically produced in glands, plant hormones are synthesized in various tissues and can function locally or be transported throughout the plant. The interaction between different phytohormones are highly complex, often involving synergistic or antagonistic relationships, then fine-tune plant responses. Understanding these plant hormones and their role is fundamental to developing effective methods for their extraction and quantification. Next, I would like to present that phytohormones exhibit a wide range of chemical structures. They can be broadly classified based on the biosynthetic origin. Most of them are derived from three key metabolic precursors. The first is amino acids, the second isoprenoids, and the third lipids. Amino acid-derived hormones include auxins, melatonin, ethylene, salicylic acid, and polyamines. Isoprenoid-based hormones, there are such as cytokines, brassinosteroids, gibberellins, abscisic acid, and strigolactones. Lipid-derived hormones such as yasmonic acid are synthesized from alpha-linoleic acid, and they primarily, for example, involve indifference responses and wound healing. Each of these hormone groups has unique biochemical properties and regulatory function. This diversity makes their extraction and quantification particularly challenging, as different methods must be tailored to the specific chemical nature of each phytohormone. With this understanding of the biosynthetic origins, we can now explore how specific metabolic pathways contribute to the production of different phytohormones in plants. They, of course, are formally distributed throughout the plants. They synthesized and transported to specific tissue. This distribution depends on development stage, environmental conditions, internal signaling networks. For example, auxin I primarily synthesized in shoot apical meristem and young leaves. Cytokinin is produced in root tips and travel upward to promote, for example, cell division, shoot formation. Gibberellin are formed in growing tissues, such as young leaves and developing seeds. Ethylene is the smallest molecules, gaseous hormone produced in response to stresses. The next, for example, strigolactone are produced in roots and help regulate shoot branching. Understanding where these hormones are localized is critical for choosing the right extraction methods. Since the distribution varies across different plant organs and even with specific cell compartments, sampling extraction techniques must be carefully designed to target the precision location of each phytohormone. Over the years, scientists have identified 12 major classes of plant hormones, each with a distinct chemical structure and biological function. This includes auxin, cytokines, brassinosteroids, salicylic acid, and other 2 peptide hormones. Each phytohormone is structurally unique, ranging from simple molecules like ethylene to complex polycyclic structures as gibberellins and brassinosteroids. This diversity in structure and function means that the single reaction extraction methods cannot be universally applied to all phytohormones. Instead, customized strategies must be used depending on the hormone's chemical nature, polarity, and cellular localization. We know that phytohormones can be categorized based on their chemical nature and solubility. Auxin and melatonin are indole derivatives, making them relatively hydrophobic and requiring organic solvents for extractions. The next phytohormone, abscisic acid, belong to the sesquiterpene family, while gibberellins are tetracyclic diterpenoid acids, both of which have moderate polarity and required mix of organic and aqueous solvents for efficient extraction. On the other hand, cytokines are adenine analogs, meaning they are relatively polar and regulate aqueous-based extraction methods. Brassinosteroids are polyhydroxysteroids, making them structurally similar to animal steroids. For example, estradiol, estrogen and androgens, sex hormones. and requiring lipid-based extraction methods. Yasmonates as fatty acid derivatives, making them hydrophobic and requiring organic extraction approaches. Ethylene, as I said, there's simple gaseous molecules, presents unique challenges for extraction, requiring tapping techniques rather than conventional liquid phase methods. Strigolactone are terpenoid lactone, making them slightly hydrophobic and requiring careful handling during extraction. Generally, auxin, gibberellin, salicylic, and yasmonic acid are acidic, cytokinin alkaline phytohormones. Thus, the diversity of chemical structures and properties of different phytohormones make the specific extraction, purification, and quantification, as well localization procedures necessary. This slide shows different structures of phytohormones. What about history? In the early stage of phytohormone research, bioassay, biotest were commonly used to estimate hormone levels based on the physiological effects on plants. This included methods like, for example, the Avena test for auxin or seed germination bioassay for gibberellin. Why bioassay provides some insight into the presence of hormone. They lack precision and specificity, making them unreliable for detailed quantitative analysis. Later, immunoassays such as ELISA introduced a more quantitative approach by using antibodies to detect specific hormones. However, these methods were still limited by the specificity of the antibodies used, leading to potential cross-reactivity and errors in hormone quantification. A major breakout in plant hormone analysis came with development of chromatographic techniques. High-performance liquid chromatography, HPLC, became a widely used tool, allowing scientists to separate and quantify multiple phytohormones with much greater accuracy. The next techniques, gas chromatography, particularly coupled with mass spectrometry, provide even better sensitivity and specificity, especially for volatile hormones such as ethylene. Over one of the main limits of GCMS is that the certain hormones required a derivatization step to make them volatile enough for analysis. The extra step increases processes time and introduces potential sources of errors. Over the past few decades, the field of phytohormone analysis has undergone significant advancement. Different analytical techniques have been developed, each building the previous method to improve sensitivity, accuracy, and efficiency in hormone detection. In 1980s, GCMS became powerful for analyzing volatile phytohormones. The next method popular in 2000 was LCMS techniques. Unlike GC, gas chromatography, liquid chromatography does not require derivatization, making it a faster and more versatile options for analysis different classes of phytohormones. Introduction of tandem mass spectrometry further enhanced the accuracy of phytohormones detection. These techniques allow to greater sensitivity, enabling researchers to detect more hormone at extremely low concentrations, sometimes down to the picomolar range. It also improved specificity by distinguishing between similar molecules, ensuring that plant hormones and their metabolites could be measured accuracy. One of the major challenges in phytohormone analysis arise from the complex of plant extract. Unlike simple chemical solutions, plant tissue contain a vast array of biological molecules, for example, pigments or carbohydrates. These substances can interfere with the detection and quantification of phytohormones, making a specific extraction and purification method necessary. A key difficulty in phytohormone analysis is that endogenous phytohormones are present at trace levels, often in the range nanogram to picogram per gram fresh weight. However, these hormones exist in mixture with the hundred of other plant compounds. This is a problem to resolve. Another challenge is the chemical diversity of phytohormones, like auxin and gibberellins are acidic, whereas cytokinin are alkaline. Some hormones are hydrophilic and dissolve easily in water, while others are lipophilic and require organic solvents for effective extraction. This diversity makes it impossible to use a single universal method for all phytohormones, requiring multiple approaches tailored to the specific chemical properties of each phytohormone. In this slide, I present the graph on which concentration and the properties of plant hormones and secondary metabolites in plant. There are maybe two interpretations. The first, cytokinin and gibberellins have moderate polarity and concentration generally between from 1 to 10 nanogram per gram fresh weight. Auxin and abscisic acid, yasmonic acid, salicylic acid have lower polarity than cytokinins, but higher concentration ranging from 10 to 100 nanogram per gram. On the other hand, strigolactons and brassinosteroids are the least popular polar hormones and are found at much lower concentration from 0.1 to 1 nanogram per gram fresh weight. Secondary metabolites have significant higher concentration and cover a broad range of polarities. The second interpretation is that since strigolacton and brassinosteroids are present at very low concentration, highly scientific analytical techniques, for example, LC-MS, are essential for the quantification. What is the conclusion for challenges in phytohormone isolation? The first, low concentration in plant tissues. Required pre-concentrated steps, for example, solid phase extraction, complexity of plant matrix, co-extraction of interfering substances, for example, pigments, phenolics, sugars, and difficulty in separating hormones from other metabolites. What is the approach to solving these problems? The first, employing selective extraction methods, and the second, utilizing advanced chromatography for separation. Some preparation is of course, is one of the most critical stages in phytohormone analysis, as it directly influences the accuracy and reliability of these results. This process involves breaking down the plant matrix, isolating the target hormones, and eliminating interfering substances to ensure precise quantification. The whole analysis procedure of phytohormones based on LCMS recommended for typical plant materials can be divided into 5 stages. The first is sampling, the second grinding, the third extraction, the fourth purification, and the last detection. Proper sample preparation typically accounts for 70–80% of total analysis time. It's important. Why? Highlighting its significance in ensuring reproducible results. It keys steps such as homogenization, extraction, purification, and sometimes derivatization. Sometimes derivatization. Depending on the hormone type and the analytical method used. The main goal of sample preparation is to decompose the plant matrix, making the phytohormones accessible while preventing degradation. Given the complexity of plant tissues, techniques like solid phase extraction or liquid extraction are commonly used to concentrate and purify the target hormones before final detection. The accuracy of phytohormone analysis begins with proper sample collection and storage. It is important since plant hormones are highly dynamic and can rapidly degrade or change in concentration due to the environmental factors. Carefully handling is essential to maintain these original levels. When collecting plant tissue samples, rapid manipulation with minimal disturbance is crucial. Even minor stress, such as cutting, can trigger hormonal changes within seconds. Therefore, sample must be collected quickly and processed under controlled conditions to prevent alteration in hormone levels. After harvesting, plant material are typically flash frozen in liquid nitrogen to stop enzymatic activity and prevented hormone degradation. The sample are then stored at -80 Celsius degree until processing. For certain phytohormone storage conditions must be optimized further. For example, some hormones degrade in light and require dark storage, while others are sensitive to oxidation. In order to calculate plant hormones concentration in plants accurately, the weight of plant materials need to be recorded exactly. In different methods of quantification, for example, auxin or cytokinin, abscisic acid, both dry weight or fresh weight are used to express plant hormone concentration in plant tissues. For fresh weight values, frozen fresh tissue can be weighted, while for dry weight values, the tissue can be lyophilized and then weighted. Once plant samples have been properly collected and stored, the next crucial, the next important step is homogenization. This process involves breaking down the plant tissue to release phytohormone, making them accessible for extraction. Proper homogenization ensures that hormones are uniquely distributed within the sample and improves extraction efficiency. To prevent degradation, homogenization is often performed under liquid nitrogen. Sometimes we use for larger plant sample homogenization using a mortar pestle, but sometimes special homogenizators. For small amounts of tissue, micro-homogenization techniques such as vibrating ball micromills is used. Success of any phytohormone extraction process depends on efficient homogenization. Once the plant tissue has been fully ground and cell walls broken, the next step is extraction of phytohormone into a suitable solvent, ensuring optimal recovery for the next analysis of the next detection of plant hormones. The extraction is essential for isolation target hormones with complex plant matrix and transferring them into a liquid phase for next purification and detection. Phytohormone extraction is challenging due to the low concentration of these compounds and the presence of numerous interfering substances such as what I said, pigments, proteins or sugars and other structural components. An ideal extraction method must be selective, efficient and capable of preserving hormone stability throughout the process. One of the most widely used strategies is solvent extraction, where a carefully chosen solvent dissolves the phytohormones while minimizing the extraction of unwanted compounds. The choice of solvent depends on the chemical properties on the target hormones. I said on the beginning lecture about it. Hydrophilic hormones like cytokinins are best extracted using aqueous solvents, while lipophilic hormones such as yasmonate or abscisic acid required organic solvent like methanol, acetone or chloroform. In many cases, a mixture of solvent is used to optimize extraction efficiency and improve hormone recovery. Depending on different extraction modes, this method can be divided into classic and micro extraction methods. Both classic extraction and micro extraction methods are based on liquid or solid phase extraction. Classic extraction methods provide no complete extraction efficiency. Compared with classical extraction methods, micro extraction have advantages of having high extraction efficiency and miniaturization of device enrichment capacity, making them more suitable for the analysis of phytohormones. The extraction and cleanup techniques for trace compounds analysis include liquid extraction, liquid phase micro extraction, dispersive liquid micro extraction, polymer monolith micro extraction, and etc. etc. Compared to the traditional liquid-liquid extraction methods in which analytes are divided between different solvents, solid phase extraction is becoming the preferred purification techniques used in phytohormone quantification for its advantages, saving time and solvent and its higher recovery and through food. We know that solid phase extraction is one of the most effective techniques for purifying phytohormones before analyzing. This method allows for the selective isolation of target hormones while removing interfering compounds. SPE, solid phase extraction, works by passing the extracted sample through the cartridge filled with a specific sorbent material. Depending on the chemical properties of the phytohormones, different types of cartridges are used. For example, C18 for nanopolar hormones, or anion cartridges for acidic hormone. Cationic and anionic exchange cartridges are used. The retained hormones are then eluted with an appropriate solution, resulting in a purified extract ready to detection. SPE offers several advantages over traditional liquid-liquid extraction, including higher recovery rates, reduced solvent use, and improved reproducibility. This technique is now a standard step in phytohormone quantification, ensuring more precise and reliable results in phytohormone research. Along the organic solvent tested, 80% methanol has become a preferred solvent for relatively good extraction efficiency for the most phytohormones. This table shows a comparison of solvent efficiency, which is easy to interpret. In addition to a single solvent, mixtures comprising different solvents has been frequently used. For example, the modified Bialystok solvent, methanol forming acid, water in range 15 to 1 to 4 showed better extraction efficiency than a single solvent for simultaneous extraction of multiple phytohormone categories with inhibition of enzymatic degradation. The next step in this procedure is ultrasonification, which is a powerful technique used to enhance the efficiency of phytohormone extraction. This method applies high-frequency sound wave to break plant cell walls, allowing for better release of intracellular hormones into the extraction solvent. One of the main advantages of ultrasonification is its ability to reduce extraction time while improving hormone recovery. It also enhances mass transfer, making it particularly effective for extraction hormones through tough or fibrous plant tissue. Additionally, ultrasonification minimizes the need for harsh solvent and high temperature, which helps preserve the integrity of high sensitive phytohormones. Because of these benefits, this technique's ultrasonification is widely used in modern phytohormone analysis to improve both efficiency and accuracy. Of course, a few types of ultrasonification exist. For example, ultrasonification assisted extraction, microwave assisted extraction, vacuum microwave assisted extraction, and other. In addition to conventional solvent extraction methods, advanced techniques such as microwave assist extraction or vacuum have been developed to improve the efficiency of phytohormone isolation. Both methods offer faster, more efficient and environmentally friendly alternative to traditional extraction methods. These techniques are particularly used when dealing with complex plant matrix or when working with delicate, sensitive phytohormones that require gentle handling during extraction. Liquid-to-liquid extraction is, of course, used method for isolation phytohormones from plants. This technique is based on a principle that solvent can dissolve itself between two solvents, typically aqueous or organic phases. By carefully selecting this solvent, phytohormones can be efficiently separated from plant components. This extraction, this method, is particularly used for removing unwanted polar or non-polar impurities. Hydrophilic phytohormones such as cytokinins are extracted into aqueous phase, while lipophilic into organic phase. Now, some improved liquid-to-liquid extraction methods are developed to simplify sample preparation procedure, including single drop microextraction, wetting film extraction, homogeneous liquid-to-liquid extraction, and other. Solid phase extraction using cartridges filled with matrix compounds, which extract compounds based on diverse interactions, including adsorption, hydrogen bonding, polar and non-polar interactions, cation or anion exchange, or size exclusion, have been widely used for high throughput phytohormone extraction and purification. Many SPE columns, such as SEPAC C18, Waters Oasis HLB, Oasis MCX, and Oasis Max, containing different sorbents, are now commercially available for phytohormone purification from crude extract, so that SPE has become the mainstream method for purification of phytohormones. I like this column, I like these methods, of course. Mixed mode, reverse phase cation exchange, anion cation cartridges are commonly used to purify acidic plant hormones like brassinosteroids, leveraging the two types of interactions to enhance the sensitivity of LCMS-MS quantification. Another type purification methods employing C18 and the mixed mode sorbents are also utilized to quantify multiple phytohormones simultaneously. For example, auxin, jasmonic, salicylic, abscisic acids, gibberellins, cytokinins, brassinosteroids. Another type of extraction method is solid phase micro extraction, which is more recently developed for SPE then use fibers coated with an extracted phase, which can be either a liquid polymer or liquid sorbent. These techniques enables the extraction of volatile and non-volatile analytes from various media, including liquid and gases. SPME offers the advantage of high recovery rate, greater than 80%, combined with simple and efficient operation in extracting and purifying phytohormones. The next extraction techniques is a magnetic solid extraction. Is an improved SPE for enrichment and purification of target analytes from large volumes based on the use of magnetic. In this procedure, a magnetic sorbent is added to a solution or suspension containing the target analyte. The analyte would be adsorbed onto the magnetic adsorbent, and then the adsorbent is adsorbed, analyte is recovered from the suspension using magnetic separator. The slides show this, this procedure, and after extraction, the next essential after extraction, the next essential step in phytohormone analysis is purification. Since plant extract contain a complex mixture of compounds, purification is necessary to remove interfering substances that could affect the accuracy of hormone quantification. One of the most commonly used purification methods is chromatographic separation, including thin layer chromatography, TLC, immunoaffinity chromatography, or high performance liquid chromatography, HPLC. These techniques allow for the selective isolation of phytohormone based on their chemical properties. Purification not only enhances detection sensitivity, but also improves the stability and reproducibility of this analysis. Without proper purification, contaminants in plant extract could interfere with mass spectrometry or chromatographic separation, leading in inaccurate results. Once purification is complete, the final step in the workflow is detection and quantification, which provides precise measurements of phytohormone concentration in plant tissue. Purification and the cleaning crude extract from plant sample can remove the bulk of interfering compounds and improve separation by HPLC and detection by MS. The choice of purification methods depends on not only the target analytes, but also on the type of analysis to be performed, as well as it's very important for us, the available analytical instruments. In addition to these methods, such as quick, easy, cheap, effective, large, and safe catchers, and dual cloud point extraction are also explored in phytohormonal extraction and purification. What is important sometimes to use is derivatization. This process involves chemically modify the hormone molecules to enhance their stability, detectability, and volatility, making them more suitable for analysis. Derivatization is particularly important in GCMS, where certain phytohormones such as auxin, abscisic acid need to be converted in more volatile forms. Common derivatization techniques include methylation and silylation, which improve the separation and the sensitivity of these compounds during analysis. Stabilizing the target hormones and improving the chemical properties, the derivatization ensures that even low concentration phytohormones can be detected with high precision. This step is essential in case where direct detection methods do not provide adequate sensitivity or selectivity. We must remember about GCMS, or when we detected phytohormones using by LC-QTOF MS. This slide is the last slide in my lecture. The final step, detection, I would like to present in the lecture hormone profiling in plant tissue. Thank you very much for your attention. You must the phytohormone detection is very easy, but we must have a very good procedure to isolation. From extraction, purification, and other steps must be complete. If you have any questions, I suggest write me on my email, and I try to very quickly answer on the questions. If you not, Thank you very much. Thank you.
Thank you, Professor Andrzej, for your nice lecture. So, the next lecture will start at 3:30, and that is basically a hands-on training on water potentiometer, and it will be by Dr. Yamshi Arif. Okay, just wait for a few minutes, then we have a hands-on training inside the lecture theater on the water potentiometer.
potential meter. So before discussing about or before learning about this water potential, do you have any idea that what is water potential? Any idea? So basically, water potential is the energy which is exerted by water or any biological system or any sample. So basically, water potential is denoted by letter psi. This water potential is the movement of water in the system. This psi of water potential is equals to solute potential plus pressure potential plus gravitational potential. So we are having this viscous water potential meter, or we can also say psychrometer. So we'll discuss about in brief, then I'll tell you step by step that how we can operate it and how to calibrate it and how we can measure reading in different biological samples. So viscous water potential is a specialized psychrometer which is used for measuring water potential in leaf disc or in any soil sample. It is basically very important tool or very important trait which is useful for researchers for measuring water potential under a stressful environment and non-stressful environment in any of the biological samples. This water potential uses thermocouple hygrometry to measure water potential or psi of any biological system or soil sample. Basically, it is designed with C52 chamber which house the thermocouple sensors and have samples housing in it. This device measures the dew point temperature of the air equilibrium with the sample from which water potential is calculated. So, the basic principle of this water potential which is denoted by psi is directly related to relative humidity which is RH. So, this psi is equal to RT upon Vm to natural log of RH, where R is gas constant, T is temperature and Vm is volume of water, and this RH is relative humidity. So components of this water potential, as you can see, this is the main unit of water potential. Here we are having this display where we can record or note the readings. Here we are having this electronic devices where we can assemble all our chambers and wires. These are basically the sample chambers, or we can say C52 chambers. In these C52 chambers, we are having sample holders. In this sample holders, we will place our leaf disc or any of the biological sample or soil samples. We are having, these are basically thermocouple sensors. Basically, these sensors do sense that wherever water vapor, or we can say these sensors do calculate the water potential, that what is the dew point of this particular leaf disc or any sample. These are the cleaning solution and calibration solution which which we are using to clean the solution and calibrate the solution, calibrate our water potential. So working principle of this water potential, we are having Vesta water potential. So it operates basically on the principle of thermocouple hygrometry. So what is this thermocouple hygrometry? This is a technique basically which measures water potential by assessing the relative humidity and dew point depression in a sealed C52 chamber. So the process begins with a sample preparation where a small biological or soil sample such as a leaf disc or stem segment or soil pores are prepared, and they are placed under C52 sample chamber, and these are specialized airtight compartments. I'll show you this how this airtight compartment works. Just this is the basic principle of it. So once the sample is inside this chamber and the chamber is sealed, initiating the equilibrium phase. So what is this equilibrium phase? Basically, during which the water in the sample exchanges moisture with the surrounding air. So once water vapors are created, obviously, allowing the water pressure to stabilize at a level that corresponds to water potential of a sample. After equilibrium, what happens? Cooling phase begins. So whenever the cooling phase begins, there is an electric current which is applied to thermocouple junction inside the chamber. This causes thermocouple to cool down below the dew point temperature, leading to condensation of water vapor onto its surface, and hence the readings are calculated or noted here only. So the next stage is the measurement phase, where we can calculate the reading as thermocouple detects, and we can record the reading by, here on the display. Readings will appear and we can record it easily. This difference is direct function of sample's water potential, as the relative humidity inside the chamber is governed by moisture content of the sample. As I have told you earlier, that basically water potential is the movement of water within the system. So what water is moving, and after condensing what we are getting, that is calculated under this water potential meter. Finally, the calculation phase, the potentiometer processes these temperature readings using psychrometric equation. As I have told you earlier, what is the psychrometric equation? That is psi is equals to RT upon Vm to the natural log of relative humidity, which relates the dew point depression to water potential of segments in megapascals. So readings here are recorded basically in megapascals. The instrument provides a precise and reliable measurement, allowing researcher to assess water stress plants, soil moisture level, and overall water dynamics in any of the biological samples. So what are its applications? As you are all are doing particular this Gian course, so you all know that why you are doing this, what are its uses. So basically its application is, it is used to measure water potential in plant tissues under stressful and non-stressful environment. It determines soil moisture potential in irrigation management. Apart from this, it also evaluates seed hydration level for germination studies. It assists plant-water relation in physiological research, as water-plant relation is very much important in plant physiology and stress physiology. It also monitors water status in forestry and ecological studies. It supports agricultural research for optimizing water use. It aids soil sciences for understanding water retention and movement. It also helps in post-harvest physiology, and it is also useful in environmental studies. So its advantages are, as it provides accurate and reliable water potential. It is non-destructive method, as it preserves sample integrity. Thirdly, it is highly sensitive to small changes in water potential. Even a minute change, it can record it. So it is highly sensitive. It is portable. As you can see, I have bought it from lab to here, so it is portable device. Rapid equilibration and measurement process work across a wide range of biological samples. We can use any kind of leaf sample. It can record its reading. Apart from this, it requires small sample size. So not much biomass or not much sample is needed to record its reading or to record water potential. Helps in real-time monitoring. Means we can record, suppose I have to take readings today only. So today we can measure it easily. We do not have to wait for 1 week or 1 month to take reading. Okay? So now I am telling you step-by-step procedure how to use it. First of all, this is the main unit. Can everyone see it? Okay. So I have connected its charger. Firstly, we have to switch on it. As you all know that we have to switch on it. These are basically C52 chambers, and these are wires, thermo wires, and this small thing here is sample holder. As you can see that in this sample holder, we are having a small depression. In this depression, we have to place our biological sample or soil sample, whatever you like. Is it clear? So a thing which is important is this, that we are calculating water potential. So firstly, we should clean it. Cleaning, then calibration, and thirdly, then we have to take our sample and record the readings. Is it clear? So for cleaning, we are having here cleaning solution. This is provided by Wesker only, cleaning solution. So what you have to do here, firstly, you have to attach this thing with main unit. In this main unit, you are having 1, 2, 3, 4, 5, 6 serial wise number. So this one is chamber number 2. So you have to connect this chamber number 2 with the main unit. Then chamber number 3, you can see it. Then chamber number 3, chamber 4, and lastly chamber 5. Once these chambers are connected, you have to make sure that the sample holders are connected to it and it is sealed or not. For sealing, what you have to do, it's very easy. Just you have to slide it. You have to place your sample holder and slide it backwards. Now it is sealed. If you have to tightly seal it. If it is not tight, then reading will not come. Readings would not be accurate. So, for cleaning, as I have cleaned it already and calibrated, but I am showing you that how to clean it. Just you have to pour cleaning solution along with these small tissue papers or tissue discs. These are also provided by Weskers only. So in this sample chamber, I have placed this disc. This is the cleaning solution. You have to pour a small amount of this cleaning solution in this, and then you have to seal the chamber. It is sealed, then you have to switch on it, as you cannot see it from there, but in 10 groups I'll show you. Then you have to go to number 2 of this, and there you have to wait for at least 85 seconds for 2 minutes. Then it will clean it, and then we can further proceed for our experiment. So as I have already cleaned it, so we'll move further. What you have to do, again, whenever you are opening this sealed chamber, you have to switch off it. Or you have to switch off it from here. There are 2 options, either from here or from here, but you have to switch off it. Neither your calibration will be disturbed. Okay? So it's very easy. You don't have to take it out. Just there are these provided. You have to just insert it in it, and your tissue papers with cleaning solutions are. Now what you have to do, as I have calibrated it already, calibration is also very easy. For calibration, we are having this deionized water. Or we can also use NaCl water, it's totally up to you. We have to place this deionized water in all the 5 chambers or 8 chambers, it totally depends on you that how many chambers you are using. If you are using 5, then in 5 chambers you have to place this deionized water. Then at least for 1 hour you should wait. Now how will you know that this is calibrated? Readings will be optimized. At all level you will get same water potential reading, because you are putting same solution. You are placing same solution in all the chambers. Clear? Now moving to our experiment, as I am having 3 different kind of leaves of tomato, and similarly 3 different kind of leaves of wheat. This one is control, as it is having only distilled water. This one is hormone treated, and this one is treated with stress. Similarly, in wheat also control set or control leaf, and this one is hormone treated, and this one is treated with stress. So you all know that in stressful condition, water potential drops down. Okay, and whenever we are treating it with some hormone or with some regulator or whichever thing which is acting as stress mitigator, it increases water potential. But that water potential should be 1 or below 1. Okay, this thing is clear. So it totally depends. If suppose of control we are having water potential 0.6, and is in stress we are having water potential 0.3 say. So in hormone treated we can either get 0.8 or we can get lesser than control. It totally depends. This particular hormone treated is hormone treated plus stress. Clear? So now I have already cut a small disc of this leaf sample. So very meticulously you have to place these discs in the sample chamber. Tightly I have seal packed them. I have kept placed samples in it. Again I am switching on it. Then I am now I have basically noted here I have gone on option 2, and there we can calculate water potential. So here are different readings, but only we have to note or we have to record water potential which is in megapascals. So after 85 seconds we'll get reading of this sample 2, then of 3, then of 4, and so on. So you can come in group of 10 and record readings here. This one here is basically for water potential, megapascal for reading of water potential. Okay, यहाँ पे जो है देखो यहाँ पे सेकंड वेट कर रहे हैं, यहाँ पे जो है तुम जब नीचे करोगे नहीं दिख रहा होगा ना, बहुत लाइट दिख रहा है, ये ऊपर की लाइट बंद आ रही है। देखो अभी यहाँ पे 0 आ रही है, ठीक है, 0 इसलिए आ रही है क्योंकि सैंपल 1 में कुछ नहीं है, ठीक है, अभी आएगी ना वो, टाइम वेट करूँ। CGR प्लस स्ट्रेस वो इसके अंदर है। पे भी चेंज हो गई रीडिंग। तो दिस इज डिमार्केटिंग कि यहाँ पे जो है सैम्पल रीडिंग जो है चेंज हुई है और ये तुम कैलकुलेट कर सकते हो। यहाँ पे रीडिंग्स एग्जैक्ट इसलिए नहीं आ रही क्योंकि वाटर में डिप था, तो ऑलरेडी वाटर पोटेंशियल बहुत हाई आएगा क्योंकि वाटर में वो डिप है। अगर तुम बायोलॉजिकल सैंपल से वैसे मूव करोगे तो ये चेंज आते जाएगा। देयर इज द टाइम, यहाँ पे टेम्परेचर है, यहाँ पे तुम लोग रीडिंग्स नोट कर सकते हो। ठीक है, ये सैंपल चैम्बर है। ठीक है ना, 5 में अभी नहीं डाला। सैंपल को निकालो यहाँ से। सिंपली, और कुछ नहीं करना। ठीक है ना, क्लियर। यहाँ पे रीडिंग आएगी है ना, यहाँ पे ये भी रीडिंग है, ये चेंज हो रही है ना हर थोड़ी। देखो -2 आ गई क्योंकि, और यहाँ पे देखो यानी 3, यानी ये चैम्बर 3 की चैम्बर 3। ये यहाँ पे जो है टेम्परेचर, कितना टेम्परेचर है, ठीक है। ये आ गया तुम्हारा वाटर पोटेंशियल इज -2, ठीक है। ये WBD है, इससे हमें मतलब नहीं है, ये बेसिकली डिफरेंट पैरामीटर्स है जो कि ये निकालते हैं। क्लियर। अब बस नेक्स्ट ग्रुप। अच्छा, ये नीचे वाली ना। कंट्रोल में देते हैं जो सबका तो वाटर पोटेंशियल अलग होता है। ठीक है, नेक्स्ट ग्रुप। आइए ना, फोटो ले लेते हैं। You just have to take a look at setup. It will not get, you will not learn it only one use or just by demonstrating. Firstly, you have to learn how to assemble it, as I have already told you. Main thing is assembling. Readings which you are basically which you are noting, or which this particular water potential is measuring, is totally dependent on whatever sample you are using. Suppose I am having this stressed plant leaf, but I have cut this small slices 1 or 2 hours ago. So obviously there will be hindrance in reading. Water potential, it might give water potential reading correct, or maybe it will not give water potential reading correct, as I have already cut this slices 1 or 2 hours ago. You have to take a fresh sample of fresh leaf disc, and that leaf disc should assemble in that chamber holder. They should neither be too large or too small. देखो अब यहाँ पे 0.04 आ रही है, किसकी आ रही है, 5 की आ रही है, यानी 5th वाले की ये आ रही है 0.04, जो कि आ रही है यहाँ पे, ये जो है हाँ मेगा पास्कल। टेम्परेचर क्या है, 22.8 डिग्री सेल्सियस। ठीक है, लॉग बेसिकली ये सब क्या है, जो हमने अभी इक्वेशन पढ़ी थी जिस पे ये वर्क कर रहा है। मेन रीडिंग ये है 0 पॉइंट, देखो दे रखा है ना वाटर पोटेंशियल मेगा पास्कल, ये बता देता है। हाँ हाँ, कौन से वाले स्ट्राइक। अब ये 5th नंबर का चैम्बर है, तो ये 5th नंबर की रीडिंग है। दूसरे चैम्बर का एक बार देख लिया, खत्म। एग्जैक्टली। ये बेटा ये अलग है, मैंने बताया था ना, उसका पूरा जो फॉर्मूला होता है जिस पे वो वर्क करता है, ये पूरा वो है कि वाटर कितना है, ये पूरी कैलकुलेशन है। मेन जो ये चीज़ बता रहा है ये बता रहा है। एलएन बताया था मैंने, ये एलएन है, ये टेम्परेचर, इससे कुछ मतलब नहीं है। मेन रीडिंग ये है वाटर। ठीक है ना, अब देखो ये इसका 25 डिग्री सेल्सियस पे ऑलरेडी फिक्स है। अब मेन यही है, इसको निकालो, इसको लगाओ, मेन इसकी असेंबली है। अच्छा ये जो है इसी के अकॉर्डिंग होगा, तो सेम मार्किंग है ना, 2 3 4 5 और एक यहाँ पे ऑन होगा। ये वहाँ से स्टिकर्स लगे हुए थे, ये इतना यूज हुआ है कि ये हट गए स्टिकर, तो ये हमने मार्कर्स किए। और अपनी दूसरी जगह से कहाँ से ऑन होता है ये यहाँ पे नीचे करके पूरा, फिर रीडिंग्स हो जाएंगी फ्लक्चुएट। नीचे से यहाँ से वो करो, यहीं से ऑन ऑफ कर दो, यही सही है। इस सैंपल होल्डर से निकालो, ठीक है, इस सैंपल होल्डर को रखो, बंद कर दो, सिंपल, वो रीडिंग ले लो। ठीक है ना, फिफ्थ वाले की अब हो गया पूरा, फिफ्थ वाला आ गया, यानी अब ये पूरा एक राउंड हो गया। आ गई सही बोल रही हैं आप, कई बार नेक्स्ट ग्रुप